Hi everyone, I'm Misha and I'm a member of 2025 iGEM team from Maastricht University, Corspin. Within our project, we're introducing the iGEM interview series where we want to bring the state-of-the-art synthetic biology closer to you. In each episode, we sit down with leading scientists, engineers, entrepreneurs to explore their groundbreaking research, novel applications, and the thoughts that are shaping the synthetic biology of tomorrow. Please, let's dive in. Hello, Dr. Mason. It's truly an honor to have you as our guest today for iGEM interview series. I'm Misha, MSP student focusing on synthetic biology.
And I'm Denise. I'm also an MSP student, and I'm focusing on biochemistry mostly.
Chris,
Great. Well,
thank you so much for joining.
pleasure to be here. Thanks for having me.
So, to begin, what first sparked your interest in science when you were young?
Well, when I was actually basically in eighth grade, I learned a lot about embryology, and that was when I first learned about genetics and the fact that the first cell that we have has all the synthesis to, all the instructions to synthesize every other cell in the body, and that really it's just a question of understanding and elucidating that program that's native to every cell. Almost every cell in our body has the copy of that genetic code, but of course it manifests differently in different tissues of our body, and over time it changes. But just learning that fact that the instructions are there and we just needed to tease them out and understand them, I was and still am fascinated by that fact. And actually, at that time in the late 90s, we didn't have a whole human genome sequenced. We barely had a good map of the number of genes in the human genome. There was a lot that was left to do. So I was really in awe at the amount of mystery left at a fundamental, textbook of our life's code. So I wanted to understand it, discern it, and then understand it well enough to actually hopefully protect it, help with diseases, and then maybe even modify it as needed.
Mm-hmm.
So you're also fascinated by space exploration. Was that also a passion that was very early on, or did you develop that later in your career?
Oh, very much so. I read a lot of science fiction as a kid, Isaac Asimov, a lot of the original Foundation series, plus some other work by like Kim Stanley Robinson or other authors. So really, always had an interest in astronomy. I went to space camp as a kid, so I was very interested in space flight generally, and then also like genetics. So I've been very fortunate to combine my two biggest passions into one research laboratory.
Mm-hmm. And after undergrad, you went to Yale for your PhD in genetics, and you also combined that with Yale Law School. So our question is, what motivated you to actually blend genetics with law and ethics during your PhD training?
It was actually a lot of it was happenstance where I had been chatting with some people that were at the law school where they were looking at various intersections between biotechnology, information technology, and law, and then started reading about patents and genes. And I thought, well, that seems really odd that genes are patented, but wait, thousands of genes were patented. So I felt it as an infringement on my personal liberty that I couldn't take cells out of my own mouth and sequence the DNA and look for mutations. If I did so, I'd be infringing a patent that just seemed really wrong. So I feel like if I want to do something with my own cells or my own DNA, that should be fine to do so. But it was actually, was not allowed based on the patent. So I thought that just seemed really egregious and wrong. So then we fought it eventually, first in legislation, then eventually in court, and ended up winning. Of course, you probably know from like Supreme Court in 2013 ruled 9-0 that you cannot patent genes, which makes sense because the companies did not invent the genes; they were already in nature.
Mm-hmm. Yeah.
Okay, so going into the NASA twin study you did, you were a lead scientist in the twin study, and you were tracking Scott Kelly during his year in space. What were the most striking changes you observed in Scott's biology when he returned to Earth, especially compared to his twin?
Some of the biggest changes were the gene expression differences. So thousands of genes were rapidly changing their expression, their activation or deactivation upon landing, which we've since confirmed in other missions and other crews. And then also his telomeres got a bit longer, which worked with Susan Bailey. That was surprising because we thought maybe they'd get shorter because longer telomeres are a sign of youth generally. So that was a surprising result. And then we also, saw, microbiomes a lot of microbiome change, like his microbiome change in terms of a response, but it went back to normal. A lot of us are lipids changed, proteins we saw change. So a lot of his body adapted and changed, and metabolites, but they unfortunately almost all went back to normal after the mission. So the body was extremely plastic and responsive. But actually, the other striking thing was the day he landed was even much more impactful on his body, and I think much harder. So I'd like to say sometimes that going to space can be hard on the body, but coming back from space is even harder in terms of adaptation. But even there, the vast majority of those big changes within a few days resolved, or at the latest in a few weeks. And so we saw that the body has a really strong resilience to the stressors of spaceflight.
Just out of interest, what kind of samples do you harvest for these kinds of things?
A wide range. So it's blood samples, urine, stool, saliva, swabs. We take anything we can from anybody we can. So like every cell tells a story. We're here to listen, whether it's human or microbial or otherwise. So we yeah, it's many, it's thousands of samples, but it's swabs, saliva, it's stool, plasma, sorted
Okay,
cell populations, many
so
kinds of cell
everything you
types.
can get.
Yeah, exactly.
Nice, nice. And you've also pointed out in one of the interviews that today humans are too frail to survive very long space journeys. So, in your opinion, what is the biggest biological obstacle for now that we need to overcome to make the long-term space journeys or maybe living in space possible?
I think radiation is the one we worry about the most, especially for interplanetary travel. When you're on a planet, you have a lot of protection. You have an entire planet between you and half of the sky, or even a moon. So once you get somewhere, you can go underground, you can build protection. It's the interplanetary space that's the most dangerous. But then once you're there, it is much more a risk of food, of nutrition, cognitive changes, just interpersonal changes, the social dynamics. We think we can engineer a lot of the risk away for food and sustenance, but Mars is an austere environment. There's not that much there. Same with the moon. So we have to bring a lot with us, and we have to really build a lot of technologies to process every atom that gets left from our bodies and reuse it. One of my favorite quotes that Scott Kelly said about the year in space mission is he was asked once and said, oh, is it true that they recycle urine and then you would drink the urine, you have to drink your own urine? And then he smiled and said, well, yeah, not just my urine, it was everybody's urine, because you recycle it all. So, but the reclamation on the space station is about 97, 98%. It's very good. So, if we can get other atoms and other molecules to be captured as thoroughly, it would be much easier to survive, in space or the moon or anywhere.
And with this, actually, synthetic biology could help a lot, I think, right?
Exactly.
Yeah. So what I heard is maybe how during these missions, if you look back, what synthetic biotechniques could have been implicated to maybe make the astronauts' lives easier.
Yeah, I break it into two big categories for synthetic biology. One of them is could you modify existing genes in the human genome to give you more of what you already have, more or less actually. So more, for example, would be vitamin C. We have the gene to make vitamin C in our bodies. We could reactivate that gene, or genes that can regulate the immune system, turn them on or turn them off to make it so you have less inflammation. Ways to kind of regulate the body a little bit better, which you can do with pharmacological agents. You can do with exercise can help, but also you could do epigenetic versions of CRISPR. You use dCas9 to just turn things on or off. So that's where you just modify the genetic catalog that you already possess. The second big area is, do you add entirely new components to human cells or to E. coli or to tardigrade genes, for example, using some of those radioprotection measures? So the second area is today very small, but I think long term will be very large. It is, how do you combine different genes together to get an entirely new phenotype or a new ability to process waste? It could be to process plastics. This idea of taking genetic elements from other organisms as tools to solve a problem, basically. And that's something that, again, is small today, but I think will encompass many, many decades and centuries of research ahead I think every time we sequence an organism, it is a potential toolkit that could be useful for a problem we haven't yet seen. And so having good annotation and characterization of many genes, of all genes from any organism, could be a clue towards a way to solve a problem, or at least a window into adaptation, just if you study it. So I think that's why I like sequencing. It's why I like genetics, and the synthetic biology is like we've been using tardigrade genes in human cells or doing engineered microbial mixtures to make whole ecosystems is a longer challenge. I mean, we're at the very early stages of ecosystem design, or even like bringing back, de-extinct species like the dire wolves, something that just came out this year, is very early days. But I think it is, the magnitude and the complexity of the problem does not obviate us from the responsibility to do it well and to do it and thoroughly, I'd say, and to do it on this planet and someday others.
So going back to humans in space, in your book, The Next 500 Years, you argue that humanity has a moral duty to spread life beyond Earth. Can you explain why you believe we need to become an interplanetary species?
My pleasure. So it is something I call as a genetic duty or deontogenic duty, like someone a being, like if you can recognize the duty, that the thought of do I have a duty almost implies that you probably have some duty. It's a variation of Descartes' I think therefore I am, or cogito ergo sum, is that if you can think about, do I have a responsibility or do I have a duty, it probably means you at least have the capacity for it, if not probably a need to act on it, just by the virtue of asking that question. So I think for life itself, though, I frame it in the context of, our duty towards life is based on the fact that we're the only ones that we know of in the whole universe that can ask the question of, can life go elsewhere? Can life go extinct? Or we know life can go extinct, different species. And because we're the only ones that understand that it is and basically creates an auto-activated duty towards preserving life. Now, that includes current life and future life, and also trying to manage ecosystems, trying to preserve life, maybe bringing back extinct life, which we just started, but that you have to do slowly and carefully. But you have to do the same thing with existing life. If you bring, like the cane toads that were brought to Australia, were meant to solve a problem with an insect, but they made a bigger problem. So there's a very mixed history of ecosystem engineering and intervention. Ben Novak has published a really nice paper where we probably have succeeded about 70% of the time. If you take a look at every example where we've ever, intervened in an ecosystem, and about 30% of the time it didn't go as well as we hoped. So it's still overall positive, but it means we have a lot to learn on, introducing species or engineering ecosystems or modifying genetic systems. We're very, very young as a species to learn about this. But again, the challenge and complexity of the problem does not obviate us from its necessity, and I think it's duty. Where if we do nothing, like say we have perfect world peace and everything's wonderful and we're all here in a billion years, and there's no crime and everyone's got plenty to eat and everyone's just, painting and hanging out and writing books and just, chatting, and there's no everything's perfect. If we're still here and everything's perfect, everything dies when the sun engulfs the earth. So I think about this almost every morning, is that we cannot just by cosmic necessity stay here forever. That if the earth is the only home we've ever known, but if it's the only one we stay at, it'll be the only home we ever have. And so we want to and need to at some point look out to the solar system and go farther, and use everything we have learned here to get us there. So I think that's my it's just a cosmological fact that I think of it and realize it and then act on it.
Yeah.
But I don't find it depressing. It's just a fact. It's just like,
Yeah.
like if there's a train coming your way, you don't think, oh my gosh, I'm never gonna do anything about trains. I'm
Yeah,
doomed.
you,
You're like,
you
no,
gotta just jump it. Yeah.
you just step out of the way and then you go to the next spot. So if you see doom coming, paralysis is not the correct action. Action is what you should do to mitigate a risk or a disaster. Yeah.
Mm-hmm. Mm-hmm. So, and also going from the ecosystem in a large sense to the like to our ecosystem, to the people. So your book also outlines the 500-year like a plan with some of the phases. So, and what might be some first steps in that plan that we can actually do now?
Yeah, it is a 10-phase, 500-year plan. We just finished the first phase, which was basically 2010 to 2020. Most of it was mapping a lot of functional elements in the human and other genomes.
Mm-hmm.
The second phase is some of the early stages of genome modification and introducing specific genetic elements into new genomes. Genome synthesis is also a lot of what needs to be done now. Computational biology, or just a lot of the AI tools for designing genomes. One interesting one recently is Evo2 was recently published to try and do a prediction of, could you design a genome from scratch? So thinking about ways to design them and then high throughput testing, kind of like what you'll do for iGEM. When you come up with a construct or a modified system, you want to test it. And so something people are worried, like, is AI going to take all of our jobs? It'll take a lot of jobs, but at some point you have to test your predictions and we don't yet have fully AI-driven labs that test every idea. That is often something you're at the bench on nights and weekends and testing and optimizing and improving.
Yeah.
And it's just the hard, quiet work of scientists testing ideas. That has to continue for years and even decades into the future. But in the long run, I think there will be some tests, in say 20, 30 years of modified systems that would work on the moon for complete, say, atomic capture and recycling, for potentially slight modification for radiation protection if there's a medical need. That's one of the things I really point out a lot, is that there has to be an unmet medical need for you to do any genetic modification for any ecosystem or for any human or for anything anywhere. If there's no need to modify it, if the system is already adapted, then you don't need to change that much. But if we start sending everyone to the moon and they're all dying from excessive radiation, we have to start to get creative.
So, genetically editing humans raises a lot of ethical questions. How do you balance the ethics of potentially re-engineering human genes for survival?
It's easy. If it took everything from a 1 billion year time frame, then it becomes quite clear. I think if you, again, the first metric is there an unmet medical need? And if the answer is no, then there's not much to do. If the answer is yes, like in the sense that we see people are dying and we have not found a way to save them, but we think we have a way that might involve genetic modifications, well, then there's a case to be made. I think the best example is He Jiankui, who did the CRISPR babies in 2017, 2018, when all the news came out.
Yeah.
He was trying to make it so that the embryos could not get infected by HIV, so it deleted the CCR5 locus, which in his mind was, he was thinking, well, there's these parents who've had HIV and they want to have a baby that could never be susceptible to the disease. So that seems logical. It's logical and seems almost reasonable. But then you have to put it in the context of, well, did that solve an unmet medical need? And the answer is no. Like, there's ways to give birth to children where they don't get HIV. There's treatments for HIV that are very effective, that don't involve, large scale new clinical trials or any changes. So there was no unmet medical need that he was solving when he did that. He was just doing an experiment. And also, we now know enough about CRISPR. It actually was almost certainly off-target effects. It was almost certainly a worse way to do genome editing than what we have today, like with prime editing and other methods. So it was a fool's errand. But it's one of those moments where you can get really excited about the technology and just want to try something because you can. But if it involves human subjects, you have to always say, is there an unmet medical need that we're addressing? And if the answer is no, then you probably should do it. I mean, some people will do weird things like that. There was one researcher who just started injecting himself with CRISPR for no reason, and some people will do crazy things. But if you want to do it as a real therapeutic or really, safely, you have to ask, do I need to do this first? And then when you do, then you look at the methods and the guide RNAs and the targets. But I think, we published a paper last year looking at all the ways you could improve bone function, immune function, radiation response, for protective alleles for spaceflight, which is interesting to think about. But I think we still have a few decades of getting CRISPR methods even better, and then finding out when and where we need to apply these specific changes. And ideally, for technologically, get them so we only modify specific cell types, which we're still working on with lipid nanoparticles or other tools as well.
Nice. And also, going back here on Earth, you also did a mapping of the normal human virome. So essentially, the viruses that that live in us and that don't cause harm. So why did you decide to focus on these non-harmful viruses, let's say?
Well, another case where there's just so much we don't know. So we know a lot about infectious agents, of course, COVID, for example, we all learned a lot about recently. But there's rhinovirus, there's noroviruses, there's many flu strains. But we know very little about the transmission of normal microbes, particularly viruses. That was the idea for the Human Microbiome Project, was just to map what's normally there so we could learn from it. And we're doing that now with viruses to just better understand, who's there and what are they making, I like to say, like what organisms are around us. And are any of them mediating, for example, the gut-brain axis? Are they mediating eczema or changes in the skin or inflammation? Most of it we just don't know because we haven't had the capacity to map the viruses until really recently. So we're now mapping, human physiological data with viruses as they move in and on and around us. They're always in motion, and they're always, little passengers with us.
Yeah, and also like we heard a lot of I myself read a lot of papers
they were saying that there's currently no standard, so to say, healthy microbiome. But like, can we say that there's a healthy virome or healthy microbiome actually? Like, or maybe are you now in any kind of projects that are doing some steps to make it possible?
Yes, that's the kind of the goal of the Human Virome Project, which we just started in our lab with many others, with Mike Snyder and Ami Bhatt and many others at the NIH, the National Institute of Health. So what we know, there's ranges of microbial species, bacterial species in particular, that you can expect to find in the gut and stool that are, there's no perfect microbiome. Although maybe Donald Trump would say he has a perfect microbiome, I would guess. But officially, there's no perfect microbiome. But there are ranges, much like when you get your blood work done for cholesterol, for red blood cells, white blood cells. It's usually within a range. And so what we've defined for a lot of the microbial bacteria is what are normal ranges you can see in your gut or in your mouth, for example, that are expected species or commensals. But sometimes you don't have them at all. Sometimes you have more of one for the other. So we have that a little bit for the microbiome for bacteria, but we're just now, I think, mapping that up for the viruses, which is the goal of that project, is to find sort of the healthiest may be strong, but at least the average or the median microbiome, let's say, or virome. Yeah.
So you've also been mapping the microbes in cities. One study found about 12,000 new species of bacteria and viruses by swabbing public transit systems. What was the most unexpected discovery from that?
Yeah, it was the fact that almost everywhere you go, there's new species. A lot of the DNA mapped no known species. So there's just so much left to discover. I also enjoyed that you could see where you were in the city based on the DNA left behind. So you could see if you were near the park, you'd see more plant DNA, for example. Or you could also recapitulate the United States Census data. So whether you're Black, white, Asian, Hispanic, other ethnicities, the neighborhood you were in, the human DNA left behind recapitulated the people who live in that neighborhood. So it was also kind of interesting just to see that as well. I think so. There was, a lot of surprises there. There were also just, the seasonality we could see. We could see a lot of changes within one day at a specific subway station. So yeah, we just got 10 years of sampling. So we're in the middle of organizing the samples to have a 10-year view of 222 cities for the MetaSUB consortium. So it's really I wouldn't have thought 10 years ago that so many people would want to go out and help swab cities, but it is a very fun project just to go out and swab something. And before the pandemic, it looked really weird to be out in a public area swabbing. People are like, what are you doing? But afterward, people are like, oh, that's fine. There's someone swabbing in a public surface. They don't even look at you twice after COVID. So that has normalized our behavior of swabbing public environments.
And also has this research affected you personally? How you now treat the cities? Maybe you wash your hands more or something like that, or
Good question. So I actually do. I guess wash my hands weirdly about the same amount. If I touch a pole, a subway pole, and it's like sticky or moist or gross in some way, I'll wash my hands. But if I just touch it normally, I don't always wash my hands. But usually, I wash my hands a lot generally before the study. So I guess it was not too much of a change from my prior behavior. But if anything, I got more confidence that even if I don't wash my hands or I forget to, it's probably okay. Generally, most of what's there is mostly like shaking hands with other people. So it's human commensals. So I feel a little bit more calm about what's on the surface.
By analyzing city microbes, you can identify what city a person actually lives in.
Yeah.
Could this kind of microbial fingerprint also have applications in forensics and public health?
Good question. So yeah, we did some of this. We published some proof of concept work on this in 2021 and 23 of showing that you could infer where someone's likely from, get to about 80 or 85% accuracy of if you give me your shoe, I can tell you what city you're likely from. And so that if you combine this with other data, with mobile data, background history, any other information, you could pinpoint someone's recent history pretty well, we think. So it's the very early stages of that. Interestingly, as more people travel around the world, it gets harder to maintain these city specific traits. But it's actually, if you think about it, though, it is just a microbial reflection of what we already see at the macroscopic level, that there are, there's more, if you look for kangaroos, you're going to more likely to find them in Australia. And if you're looking for, lions and tigers, you'll see them more in sub-Saharan Africa. If you're looking for, Tasmanian tigers, you'll see them now more in museums in Tasmania. But, there is specificity for macroscopic life, and so we just see that reflected also microscopically.
Mm-hmm. And also now talking more on the entrepreneurial side, you co-founded Nurture Genomics to bring the whole genome sequencing to newborns for guiding their healthcare, for personalized healthcare. What kind of benefits do you see for families from doing it?
Is early detection is really powerful medically. So if you can actually see different mutations in genes that you can intervene on, that you can actually have a therapy that could help cure a disease, or at least get an intervention, say for PKU or other diseases where there is an easy intervention that can help save a life. And that was why we started the company. Ideally, I'd like everyone's genome to be embedded as part of their medical care, a genome guided medicine. And we're starting to get that way with cancer, but we haven't yet done this for daily care, or say pharmacogenomics, or at birth. So that's part of the reason we started the company, was to build that.
Mm-hmm. And also, yeah, a lot of people feel not very confident on sharing their genetic information. Like, yeah, usually with these, there are a lot now companies that sequence your DNA, and then they that they locate where and when you from, like your ancestry, where it is from, or maybe your predisposition to genetic diseases as well. So how do you address this ethical side of this?
Yeah, I think there's a lot of concern of when who has access to your data, who's sharing it. Like 23andMe recently went bankrupt, and a lot of that data has now been acquired by others. And I think you have to just make sure people know. Well, there's two ways to solve this problem. One is make sure people have informed consent about what they're doing when they sign up, so they know what they're giving away. The second way, which I think is even more important, is that we build the society to protect people, even in the absence of like, even if someone's consented and the data is never supposed to be released, but then there's a security breach, then that data is loose. So if we build society so that insurance doesn't discriminate against you based on your genetic code, which we have a law for that in the United States called GINA, that protects you from genetic discrimination, but we need to make it stronger and make it better, because it's only for hiring and firing and for health insurance, but it doesn't cover life insurance, disability insurance, long-term care. Now, for example, if you have Huntington's disease, which runs in families, you wouldn't want to share that information necessarily with your insurance agent for life insurance, because they could and would probably discriminate against you. So there are still some work. There's still some work to be done, but there is ways to protect, I think, both individually and at the societal level, to make it better.
Okay.
BioAstra focuses on off-planet healthcare and space-inspired medical innovation. In practice, what kind of technologies or solutions is BioAstra developing for healthcare in space?
Many things. We're working on individual sort of biotechnologies that you do, like sequencing in space, diagnostics, portable, basically detectors, kind of like tricorders if you watch Star Trek, things like that, but like real world versions of them. Also trying to get data sets that people can use and best practices for when they plan their missions, and get people prepared for, quite literally, living on the moon. We're also planning a series of other missions that would go out beyond the moon, and also have identical twins plus organoids that are miniature clones of an individual that could fly into space. So that's ongoing as well. And so it's ways to, but it's a nonprofit. We want to engage people. There's an educational component. We have training for summer students, and want to engage people to think more about upcoming missions and bold ideas for ways to have good healthcare in space, and just new technology that could help us survive out in space.
Nice. Nice. And now more on the general side, when you look at everything you do, from space genomics to newborn babies to mapping the cities, what is the unifying mission of your career?
It very much is in the book. I feel like if I'm contributing towards, like, for the next 500 years, I'll be dead for most of the next 500 years, of course, but I want the data sets and the tools and the technologies to help people survive past the life of our first sun, of our first planet. And so if the methods I've developed, if the data sets, if the technologies, if the companies help us live longer, live better lives, and have technologies that help us survive, not on Earth, not just on Earth, but also on other planets, then that is something that I am glad to do. So like a lot of the early work of just mapping viruses, for example, seems fairly mundane, but because there's so much we don't know, even simple experiments can be very illustrative as for new biology, for new components. So anytime I sequence something mysterious, I'm also thinking like, well, could that help us down the road for synthetic biology or a new function of a new gene? Because there's so much we don't know. Everything in the next 10 years is so discovery focused because we have to finish a lot of the mapping before we can do a lot of the building, I'd say.
Yeah, that's true. That's true.
This interview is part of our iGEM podcast series, and many listeners are young synthetic biologists. How do you see the role of synthetic biology in the kinds of projects you're doing?
I think synthetic biology is the natural follow-up to genetics and genomics where we can map and understand and characterize biology. But the true test, I think, of our understanding of biology is can you modify a system or even add components or subtract them and then predict what happens? Can you know what the function is, for example, of an enzyme in a cell? If you take it out, you'll understand what happens to biochemistry. Do you know the function, of a femur when you try to walk? It's much more difficult. So, it really helps with weight. There's in genetics, it's called deletion experiments, where when you remove something, you can see what happens. And I think synthetic biology is the forward projection of once you think you know what all the parts are in a genome and in a body, then the real challenge, the real test of understanding is can you change something and predict what happens and be right? And then that's on the technical understanding of synthetic biology. But then I think the even more exciting part is when you apply it and say, I want to build a microbial ecosystem that helps plants grow in lunar regolith, or I want to have an ecosystem that has a chamber that can process human waste and make it into a barbecue sauce. I don't know how that would taste, but ways where you can literally almost do alchemy, where you can convert matter from one form to the other, which is what life does every day. Like we take it for granted, but you eat food and it converts it to all the other versions of molecules you need for your body, which is kind of magical, right? And so there's magic all around us that we scarcely understand, but being able to almost create more magic is a lot of what synthetic biology can do and is doing, I'd say right now.
Yeah, that's true. That's true. And for students, as we are early on in their career, and who have also pretty broad in interest, as you did, like with genetics, law, everything mixed up, what advice would you give?
It's versus be curious and be open, and then also there's often the best thing to do is to share your data and ideas and results with people, and I think that credit is almost infinitely divisible. So collaborate with people, work with people. One of the biggest mistakes I've seen in young scientists is people who like make a bit of code and they don't want to share it because they're worried if they give away their code, then that means they won't be needed anymore. But it's actually the opposite. If you're viewed as someone that hides your data or is not easy to collaborate with or hard to work with, less people will talk to you and work with you. So I think there's some of the biggest challenges we have as a species, as a field, are bigger than one person, bigger than one lab, bigger than one country. So we have to be able to be collaborative and work with each other, and just to be to be generous with people and be open and say, you're welcome to join this project. And I always tell people that all the projects in our lab are open, that anyone's welcome to join a project, that the only limits are determination and sleep is one of the kind of the lab mottos. And I really mean that is that you can and should be able to help with anything you think you can help with, and also keep being curious, be humble, and be collaborative. Because I always learn more when I collaborate with people from different fields or different labs. I always am learning. So I think that is a great way to come up, learn new methods, new techniques, new tools, is to collaborate with people, work with different people. And it's often very interesting. So I think, yeah, and be tenacious, because a lot of times you'll send an email and it gets lost, and a second email gets lost. But really the only way things get done in general is you have to keep on it. If you really want to see something happen, often the best way to make it happen is to keep being the person that make, don't be annoying. Don't say what's up or just a question mark. Say just a gentle reminder. Be gently reminding people is the best way to make sure things get done, because everyone's busy, everyone's got a lot to do, too much to do, but you have to kind of make the future you want to see by being tenacious for it and really persistent and determined. But then also you want to be the collaborator that you would like to see in the world, which in our instance is be someone that's friendly and collaborative and open with methods and data.
Yeah.
So out of interest, what scientific breakthrough do you hope to see in your lifetime?
I'd like to see, I've seen several already that I thought would take longer, like the CRISPR systems for genome editing. I thought it would be much harder, 10 years ago, 15 years ago, that it wouldn't be as easy. But I'd like to see more of those. I'd like to see additional kinds of systems, which we've seen like already come out some of the Omega systems from other labs, but I'd like to see more and better genome editing systems. I'd like to see whole genome synthesis at scale, and tens of or hundreds of megabase scale synthesis of DNA would be great. So large scale DNA synthesis and cellular structure building. So build any kind of cell of any type that you want with any lipid bilayer profile. That would be kind of fun to see. And of course, I'd like to see people living on the moon and Mars comfortably. Having cocktails on the moon would be great.
Nice, nice. Chris, thank you very much for this interview. It was an amazing talk. We just have the last question to you.
Sure.
So, if you had an unlimited amount of resources, unlimited amount of money, an unlimited amount of time, that's important, what project would you tackle and why?
It'd probably be the same one I'm doing now, which is the mechanisms and means to get people missions that go farther into space. So we were trying to get, which are expensive than the billions of dollars, but pushing the limits of human exploration, pushing the limits of applications of synthetic biology to resource utilization and resource reclamation if you're out in space or far out in the desert on Earth, and more poetry and more art from more people, I guess, would be good as well. So more of the visualization and the appreciation of beauty as well from everybody.
Nice, nice. Thank you very much. Thank you very much for the interview.
My pleasure. Thank you.
Thanks for listening to the new episode of our interview series. If you enjoyed it, don't forget to like, subscribe, and share it with your friends. See you in the next episode.